Connectomics. So this is a cool area. Again, it's structural MRI, but it's different from what we talked about in the previous lecture because we're now looking at connectivity between different brain areas rather than these measures of sort of size, shape, thickness kinds of measures. And the history of connectomics goes back really to the 1800s, early 1800s, when Johann Riehl first published a new method for fixing the brain that allowed for the dissection of white matter tracts, because the brain inherently is very squishy and falls apart quite easily. And so being able to fix the brain and have it make it solid enough that you could actually tease apart these tracts was a critical first step. And then Karl Burdach produced a 3-volume atlas, basically, of human white matter anatomy a few years later using this technique. And then in the late 1800s, this, sort of having described the anatomy of the brain and these different white matter pathways and so on, Theodor Meynert developed the first, what he called hodological, which is just a fancy word for connectivity or something like that, theory of brain function. So sort of looking, if you think about, this was sort of in the same era that phrenology was popular and then debunked, and then Broca's work is coming out and Wernicke's work is coming out showing that different brain areas seem to be specialized for different kinds of things. So Meynert's sort of complementing that with this idea that different brain areas are connected and that brain function and cognition really rely on interactions between different brain areas, so more of a network kind of theory rather than just a phrenological area lights up, it does this thing. Karl Wernicke actually described the first disconnection syndrome, so whereas Broca described the first association between a particular brain area being damaged and cognitive deficit, Wernicke documented that there was white matter damage which basically he described as a disconnection between two different brain areas, and that was associated with different kinds of language deficits. So showing that these, sort of supporting Meynert's hodological theory. Flash forward almost 100 years, and Basser, Mattiello, and Lebihan developed and showed the first diffusion tensor imaging studies, so using MRI to look at white matter tracts. So in between 1874 and 1994, all that we knew about white matter tracts was still based on postmortem dissections and animal studies. So in animals, you can do tracer studies. You can both dissect, but you can also do things where you inject dye into a particular brain area, wait a certain period of time, then sacrifice the animal, section the brain, and look at where that dye projected to. And so the idea is if you inject it into neurons, the dye will filter down the axons to the end. And so you can basically trace out those axons and figure out where those neurons project to. Obviously very tedious, time-consuming work, but it does allow quite accurate gold standard mapping of how different axons project and how these white matter tracts go. But obviously that has limitations because we don't want to be injecting stuff into human brains and then chopping them up. So it's really limited to animal studies. So being able to do this with MRI non-invasively was a huge step forward, limitations notwithstanding, which we'll get to. And this led to the development of the field of connectomics. So connectomics is actually quite a recent word. It's become a very popular buzzword. There's like TED Talks about it and stuff. But it was really like 2005, I think, that the word first surfaced in the literature. But the idea is the connectome is the complete set or matrix of connections in the brain. And so what this figure is showing is, you can't read the labels probably, but they're different brain areas labeled along one axis. And it's a correlation or connectivity matrix. So the same areas are represented over here. And so the diagonal is all gray, and that's basically like an area is connected to itself. But every other square shows whether or not there's a connection between those two brain areas. And so the idea is you map out all the different brain areas and tell which areas are connected to each other, and that's the connectome. The connectome is presumably quite consistent across individuals because it's probably largely genetically derived. But you can imagine that there might be differences, obviously extreme differences in cases of brain damage and so on, but different genetic variants might cause differences in the connectome, as might experience. If experience strengthens or weakens different pathways, this could influence things as well. So the connectome is a complete set of connections. The projectome is another term, and actually Dr. Meinertzhagen in this department has been at the forefront of thinking about projectomics and defining that term and working with it. So it's like the connectome. The difference is that the projectome is directional connections, whereas the connectome, we don't really know. We just know two areas are connected to each other, but we don't have any idea which direction the information is flowing in. And that is one of the inherent limitations of the MRI-based diffusion tensor imaging, is that we know, or we infer that two areas are connected when we see a connection, which I'll talk about, but we don't know the direction of it. The only way that you can really get at that projectome is by doing the animal tracer studies, because you have to know where the cell body is and where the axon ends, and that'll give you an indication of which direction the information is flowing in. So some areas in the connectome might have reciprocal connections going both ways. Some might just be going in one direction, and we don't fully know that, and we can't know that from the human non-invasive imaging techniques. So diffusion tensor imaging is the approach that's used to map out the connectome in humans, and you can also apply it in other animals. And it generates probably the prettiest images of MRI or any cognitive neuroscience technique. And the reason they're so pretty is because we're trying to represent very complex 3D structures in 2 dimensions. And so the color coding is, you can actually see it over here. This one is a little less compelling than the one on the left, but it's different 2D slices through the brain. And the idea is that the colors are representing different directions. And so actually the circle is a little bit confusing, but basically in this case, the pink is indicating X, so left to right connections. Bluish purple typically represents vertical connections, and then green represents back to front. And so if you've got a connection, because obviously there aren't just straight lines in the brain, they're all sort of curved and different angles and that sort of thing. And so where you get the different, like the cyans and so on, is where these things are going at a direction that's sort of a combination of the anterior posterior green and the inferior superior blue, so it's some sort of angle. So it allows us to represent these very complex 3D structures in 2D just using the different colors. So those are just pretty representations of fiber tracts in the brain that we can get out of DTI. So that's what it is. How do we do it? Well, we use MRI. I've already said. And what we're actually measuring, and this, like many MRI measures, is an indirect measure of something. What we're measuring is Brownian motion of water molecules. And Brownian motion is just the fact that when heat is present, so anything basically above sort of, absolute zero, and certainly any liquid water, the heat energy causes those H2O molecules to move around in random fashion. So it's just this randomly occurring, very microscopic kind of motion that occurs. And the water molecules sort of float around in something like CSF. You can imagine that those water molecules can go in any direction. But in the brain, there's barriers to diffusion. In particular, the white matter has myelin, right? So axons are wrapped with this myelin sheath, which is composed largely of fat. And fat and water don't mix, oil and water don't mix. That's a nice English saying. So when the water molecules encounter myelin, they don't go through it. They kind of bounce off it or go in other directions. And so that's kind of the basis of DTI. So when we talk about this diffusion, this Brownian motion, we call it diffusion of water molecules. If it's free diffusion, so if it can go in any direction, the term for that is isotropic. So it's sort of it can diffuse in any direction. In contrast, if there's barriers to diffusion, we call that anisotropy. So that's where diffusion occurs preferentially in some directions and not in others. And so the white matter tracts of the brain provide barriers that create anisotropic diffusion. So as I said, fat is hydrophobic is the fancy word for water and fat don't mix. And so axons, it's estimated that they represent or contribute about 60% plus of the anisotropic diffusion in adult white matter. And the rest comes from myelin. So those two things are sort of related. But you can think that the myelin's around the axons, so water within the axons is going to diffuse within the axons, and water outside is not going to diffuse across the myelin, but sort of parallel to it. So we're getting information from both inside and outside the cells because of these barriers to diffusion. A couple of important points. So this is a structural imaging technique, meaning it doesn't tell us anything about function. Sometimes people get confused and think that it's actually mapping like the flow of neurotransmitters or water or something through the axons, but it's not. It's just this random diffusion that occurs. The diffusion isn't caused by any sort of physiological process. And so you can get essentially the same diffusion tensor imaging map from a corpse as you can from a live human being. There's no, sounds like fun, but there's nothing about being alive or brain function that contributes to these images. It's all about the structure. But it's unlike the other structural MRI techniques that are basically just sort of telling us, is there white matter there, is there gray matter there, is there CSF? This is giving some information about how and the direction in which water diffuses. So how do we do that? We use MRI. And we're using kind of a trick over time. And so if you think back to when I talked about MRI physics, to get an MRI image, we put the body in a strong magnetic field that causes this net magnetization vector, which is the alignment of all the protons within your body, to generally trend towards alignment with that strong magnetic field. Then we send in the RF pulse, and that energy, which is at the Larmor frequency, which is the precessional rate of those protons. So that causes the net magnetization vector to tip because it's trying to align with that RF energy, which is another form of magnetic field. And so we send in that pulse, then we turn the pulse off. The precession gradually comes back into alignment with that strong magnetic field, and we can measure the amount of energy and time that takes. So that's sort of the fundamental principle of MRI. In diffusion-weighted MRI, what we do is we send in one RF pulse that causes a 90-degree flip. So the protons sort of change direction. And then we send in what's called a refocusing pulse a certain amount of time later that kind of unwinds those effects. So this actually has a cool little animation. So we turn on that RF pulse and we're changing the alignment. And I think we click again. And then this second RF pulse should unwind or rephase the molecules. So basically you sort of flip them and then you turn them back. That's the principle. That's assuming these guys don't go anywhere, right? So they stay in exactly the same place. Because remember, everything, when we're doing MRI, everything's spatial, so everything's sort of tuned to a particular slice, and within the slice, the sort of matrix of positions within the slice. So if nothing changed, everything would line up, and you'd end up with this situation where everything's back into alignment with B0, the strong magnetic field. So we apply one that tips them, and then the other one tips them back. But of course, if the water molecules are diffusing, that's not going to happen perfectly, because as they move around, they're moving spatially through the brain. So they're moving across these magnetic field gradients that we've created. And so the Larmor frequency isn't exactly the same at each point. So when we send in the second pulse, it's not going to fully unwind the effects of the first pulse. And so you can see that here. See, these guys are moving all around. They get tipped by the vector or the RF pulse. And then the second one occurs, and you can see some of them have actually diffused right out of the slice altogether. And afterwards, we get some alignment with B0, but it's not nearly as full or perfect. And so you get effectively a signal loss, right? Because these protons are contributing to our signal to the extent that they were tuned to and affected by the RF pulse. And so in this case, relative to the case where there was no diffusion, we have a signal loss both because some of the molecules have drifted right out of our imaging plane altogether, and because some aren't perfectly rephased. And this just basically sort of illustrates that in a non-animated kind of way. Right, so before you've got alignment of the protons with B0, apply our winding gradient. Remember, we actually have a phase roll across the slice, so not everything's affected the same way, but everything's tilted to a certain highly predictable amount. And then if there's no diffusion, the unwinding gradient should bring them back into alignment. But if there is water diffusion, which tends to happen, the DTI signal is relatively weak. So that's the basic principle of this. what we actually do in practice in order to figure out which direction diffusion is occurring along is that, which this isn't really illustrating clearly, is that we remember everything with MRI again is gradients. So we can have a gradient along a particular spatial dimension. And so when we're doing this, we're inducing this phase roll along a we're inducing the phase roll along a particular dimension and then turning it off. And so what we get is a measure of the amount of diffusion along that dimension. And so we can do this across multiple sort of shots or parts of our scan in different directions and compute the amount of diffusion that's occurring in different directions. And so at each voxel, we should get differences in the amount of diffusion depending on whether we're sensitive to, say, vertical or horizontal or some angle of diffusion. So to do the imaging, so typically we're doing this in humans at like a resolution of 1 to 2 millimeter cubes. There's a real push in the MRI world to go to higher field, like 7T scanners have become quite popular for doing diffusion tensor MRI in particular, because they offer, so basically as you get a stronger magnetic field, you get better contrast to noise or signal to noise ratio. So you get less noise in your images, basically. And the big problem with improving the spatial resolution of MRI images is the voxels, the noise never changes. So if you think about sort of the amount of noise in an image, that's considered a fixed property of the scanner based on the strength of the magnet and other factors. But if your voxels get smaller and smaller, your voxels are the source of signal or contrast. And so smaller voxels mean a worse signal to noise ratio inherently because the noise is fixed, but the amount of tissue that you have generating the signal goes down. And so by improving the field strength of the magnet, you're improving your signal to noise, and so you can get down to smaller voxel sizes. And it turns out this becomes much more important in diffusion tensor imaging than it is in functional MRI, just because the spatial resolution that we need to do most cognitive neuroscience kinds of functional MRI studies is, 2 millimeters is kind of a reasonable level. But when we're talking about white matter tracts that are quite small and fine and have very complicated shapes, having that higher resolution is actually very valuable. So there's a big push there to get to like sub-millimeter resolution using these higher field scanners. And then another parameter that you set, which is unique to DTI, is the B factor, which is the strength of the diffusion gradient. So again, that's, along this direction that we're looking at diffusion, how strong is that magnetic field difference between one end and the other end of that spatial dimension. And essentially the higher your B value, the more sensitivity you have, and again, you get some sort of improvements in spatial resolution there. Then the other thing that's fairly unique to DTI relative to most fMRI is using cardiac gating, which basically means that you put some sort of pulse detection, like a chest strap or something, on the person when they're in the MRI, plug it into the MRI so that you actually have their pulse being recorded by the system, and you can time the pulses, so I'm using pulse in a different term now, so the RF pulses, so the actual MRI scanning to the cycle of the heartbeat, so that you're always grabbing the images at the same time synchronized with the person's heartbeat. And the reason that's important is because there's so much blood pressure that the brain actually pulses with every time your heart beats, and it moves it slightly. And when we're doing this super sensitive, high spatial resolution kind of measurement, those pulsations of the brain will actually cause noise and distortion in the images. So by constantly syncing the scans to the heartbeat, we get much better signal to noise and less distortion that way. And so, as I said, we need different directions. We need to measure diffusion along different directions to get the full sort of 3D picture of how the water molecules are diffusing. So typically you're using an echoplanar pulse sequence, so the same basic pulse sequence as in functional MRI, for the same basic reason that it allows you to get an image very quickly, because we've got lots of images to get in this case. So again, like DTI is not time resolved, we're not measuring things over time or changes over time the way we are with fMRI. But still, we want to go fast to be able to capture the amount of diffusion that occurs in a small amount of time and capture the whole brain in a time-efficient sort of manner. So to get a DTI scan of the whole brain with enough different directions of diffusion to make it worthwhile for the kind of work that we're talking about is probably like a 10 to 15 minute scan. So it's a relatively long scan, even using a fast imaging technique. So normally you'd acquire one or more images with a B value of 0. So again, B is the steepness of the diffusion gradient. So a B of 0 means there's no diffusion gradient. So it's kind of like our baseline or reference image. And then you get somewhere between 3 and 100 images with weightings in different directions of diffusion. So 3 is sort of the bare minimum that you get X, Y, and Z, but that's not really enough to capture all the different angles of possible diffusion. So more is better up to a point. 100, it depends. A lot of studies use more like 50 different directions of diffusion, and they're just sort of as different from each other as possible to capture as many possible different directions. Obviously, the more images, the more directions of diffusion you take, because each is an image, the longer the scan takes. And so there becomes a trade-off between, yeah, more directions would be, give us a little more sensitivity, but the person's going to have to lie still in the scanner for half an hour instead of 15 minutes, and that's just not really feasible. 15 minutes is already a pretty long time to hold terribly still in the scanner. All right, so that's how we get the images. So in terms of data analysis, some things are quite similar to what we would do with a functional MRI image. The first one, though, is a little different. So we have to do motion correction because, as I say, 15 minutes is a long time to hold perfectly still. So it's entirely likely that somebody's going to move a bit during the scan. And because we're acquiring a whole bunch of different images with different directions of diffusion, those actually come out from the scanner as different images. And so we can do the same kind of motion correction that we would do on an fMRI scan and just align those with each other using standard image processing measures. The other thing that we're doing sort of intrinsically in that step for DTI is eddy current correction. So eddy currents are these magnetic field currents that get induced by the pulse sequence that can really affect our interpretation of DTI images, in particular, the diffusion measures. So that's an important step, but that's sort of taken care of as part of the motion correction. Then we would do spatial smoothing. And again, the motivation there is similar to why we do it with fMRI, that it helps reduce the noise to some extent. And also there's this matched filter theorem that basically says that you should apply a spatial filter that matches the size of the differences that you're trying to get. And so we don't want to apply a lot of spatial smoothing, like a centimeter or something like that, because we're looking at relatively small fiber tracts. But if the changes are more than a single voxel in size, the spatial smoothing helps improve our sensitivity to that while reducing some of the noise. Correcting for inhomogeneities in the magnetic field, so that's basically the fact that I talked in the fMRI stuff about distortion as well, and susceptibility artifacts, and the fact that when you have different tissue types, especially like air and bone versus the soft, squishy, watery stuff of the brain, that can cause distortions in the magnetic field. If somebody has any like metal fillings in their teeth, that can cause some distortions as well. And so there's a step where you can actually correct for that. Spatial normalization, so again, this warp into a standard template. That we would actually apply after we do the diffusion parameters, because we want to calculate them on the person's brain in the shape that it actually is. And then we'd sort of map that out to the template brain. And then the other thing that's done in certain types of processing streams is reducing the white matter. So you can see here in this image, the white parts, the bright parts are the white matter. And then the sort of bluish parts are what we call the white matter skeleton. And this is basically an approach that's been developed because different people's white matter tracts are going to be somewhat different thicknesses, even after spatial normalization. And so if we're doing something where we're looking just sort of voxel by voxel, comparing different brains, we're going to see big differences just because people might have sort of thicker or thinner tracts. And if you wanted to measure the thickness of the tracts, that's one thing that you can look at. But in a lot of cases, what people are looking at is trying to identify sort of where the tract is, and some measures that we'll talk about in a minute about sort of what they would call the integrity of the white matter tracts. And to isolate that separate from any differences in size or imprecisions in the spatial normalization, if we just reduce the white matter to a skeleton that's sort of the core of each fiber tract, then you're sort of isolating stuff that's going to be consistently present in every person's brain. And so you get more of a pure measure of their white matter intactness or whatever you're measuring, rather than the thickness or individual variability in the size or sort of the edges of the tracts or that sort of thing. All right. So those are sort of the pre-processing methods, and then the calculations that we would apply to actually get metrics around the amount and kind of diffusion that's occurring start with the ADC, or the apparent diffusion coefficient. So this is basically sort of the difference between each image direction of diffusion and the B0 reference or sort of control image. So how much more diffusion is occurring along this particular direction than the B0 sort of diffusion in any direction. So you'd compute this for each of your directions of diffusion. So say you had 55 directions of diffusion, you'd have 55 of these maps. And then you'd combine them together, and that gives us what we call the diffusion tensor matrix. And that's sort of the first step to do additional calculations. The main one that all of this relies on, and why it's called diffusion tensor imaging, is it's using a branch of mathematics called tensor mathematics. And it's basically defining a 3D ellipsoid with 6 parameters, including 3 eigenvalues and 3 eigenvectors. So basically what it's saying is, okay, we've got many different directions of diffusion. So at each voxel, we've got, say, 55 images that tell us the strength of diffusion in each direction. And what we're doing with this tensor calculation is figuring out which direction has the strongest diffusion. So what's the primary direction of diffusion? And this doesn't necessarily have to be perfectly parallel to one of those 55 directions. The math allows you to find a direction of diffusion that's in between them. So it's combining all of the information from those 55 directions and sort of extrapolating from that to figure out where the primary direction of diffusion is. So the eigenvectors are the directions of diffusion, and the eigenvalues are the strength of diffusion in those directions defined by the 3 eigenvectors, with the first eigenvector and eigenvalue being the strongest direction of diffusion. And this is a little sort of catches people up sometimes, the second eigenvalue is not the second strongest direction of diffusion. It's the direction of diffusion that's orthogonal, so perpendicular to the primary direction, that's sort of strongest orthogonally. So you imagine that you've got, and sort of easiest to illustrate this maybe with my pointer, right? So if this is the primary eigenvector, primary direction of diffusion, the second eigenvector is going to be in a direction that's some direction that's perpendicular to that. But we don't know if it's sort of pointing, horizontally, vertically, or it's some different thing. But it's going to be defined orthogonal to that. And then the third eigenvector is defined by sort of being the one that's orthogonal to the other 3. So you end up with an XYZ Cartesian coordinate system where you've got 3 orthogonal planes that are all defined based on which is the primary direction of diffusion, and the other 2 are defined relative to that. So the primary eigenvector is the most informative because that basically tells you at that voxel how are the white matter tracts aligned, right? The other 2 are not as informative in terms of like the direction, but where they are useful is in telling us how strongly oriented the fibers might be at that voxel. So this is showing you different kinds of ellipsoids being like 3D shapes defined by these 3 dimensions. So in A over here, so the blue is our primary. I only just realized after the book has gone to press that this is wrong. Blue is actually our secondary, red is our primary. Oops. All right, so red is the primary direction of diffusion, and blue is the secondary. And you can see they're orthogonal to each other. So if these two are equal or almost equal, that defines a sphere, right? So now it's a ball, and the diffusion is sort of equal in at least two directions. If it's a sphere, it's really equal in all three directions. So that suggests that we don't actually have a strongly oriented fiber there. We might actually, this is something that you might find more in the CSF or maybe in the gray matter. And then as you have an increasingly sort of bigger ratio between the two, you get something that's more cigar-shaped or ellipsoid-shaped like that, to the point where this is where you're starting to see, so they're anisotropic and strongly oriented in a particular direction. And so where that secondary and to some extent the third eigenvalue are informative is in telling us, is this voxel actually likely to be a white matter tract, a fiber, or is it just something else, right? So you get a combination of information both around the direction, which is really the primary eigenvector, and also the extent to which it's likely an oriented fiber as opposed to blob or something else. So these things are both useful calculations there. And so you're doing this at every voxel in the brain. And so you're going to get out of your calculations a number of measures, right? Because you've got each voxel has, primary, secondary, tertiary eigenvectors, primary, secondary, tertiary eigenvalues, and you can use these to define these ellipsoid shapes. And software packages will actually allow you to visualize the shape of the ellipsoid at every individual voxel. And so you can use these, especially really the primary eigenvector, to define these kinds of maps that I showed you earlier, which are color-coded with respect to the orientation, which is primarily based on, as I said, the primary eigenvector, but also constrained by the ratio of the eigenvalues, right? So we're going to eliminate from drawing these maps anything where we end up with like a sphere, and just take the ones that are more strongly oriented. As I said, you can visualize this in different ways. So this is one, these aren't quite sort of cigar-shaped things, but they're basically representing the ratios of the different eigenvalues. So you can see both the orientation of different fiber tracts and where there's sort of strongly oriented fibers as opposed to things that aren't terribly strongly oriented in any particular direction. You can also overlay just little red lines where the length of the line represents the strength of the orientation, and then the direction represents the direction, overlaid on a grayscale like anatomical MRI. And so here this is zoomed in, but this is part of the corpus callosum here, the white part. And so you can see the red lines there in particular sort of line up nicely with what you would intuitively expect those white matter tracts to be doing, and other kinds of visualizations. So again, lots of different ways to visualize it. Another metric that you can get out of your DTI images is fractional anisotropy. This doesn't actually tell you anything about the directionality of the fiber tracts, but instead it's just a value that ranges from 0 to 1, where 0 is isotropic and 1 is anisotropic. And so it basically tells you, if you map it out, as you're seeing in the image here, the ones that have a value of 1 are white, so they're highly anisotropic. So those are voxels where there's likely a very strong white matter tract running through them. And the black areas are the ones that have highly isotropic voxels, so likely not having a strongly oriented white matter tract running through it. So this kind of looks like a structural MRI image, and it's an MRI image and it's structural, but it's derived from computing the anisotropy at each voxel rather than just looking at like the T1 weighted anatomical image, even though the white matter appears bright there as well. Here the brightness is reflecting the strength of orientation in a particular direction. And the nice thing about this last bullet point is that where we've computed 6 values, so 3 eigenvectors and 3 eigenvalues per voxel, reduces it to a single number. So in terms of something like if you want to do stats, now you have 1 number to worry about instead of 6. So it's kind of convenient in that way. And people often use this as their primary outcome measure in DTI studies. I find it a little bit problematic because it's measuring the level of anisotropy, and the interpretation that's often applied to it is white matter tract integrity. So the idea is that, in all of us in this room, hopefully our brains are fairly intact, so we'd all have pretty high FA within. Nathan's looking around as if like, what about her? So everybody would presumably have high FA in all of their white matter tracts. That's fine. But you imagine like a degenerative disease or something like a concussion where you think there might be some shearing of white matter tracts. You could imagine that FA values would drop in damaged areas because the anisotropy might be worse. The problem with this, and this goes back to sort of big picture thinking around what we're doing with DTI, we're measuring water diffusion. And we know that things like axons and myelin present barriers to diffusion that lead to these sorts of oriented maps. But when we see a drop in sort of the strength of diffusion along one direction, we can call that white matter intactness. And if the white matter was broken down, like if the myelin's degenerating because of multiple sclerosis, or if there's physical shearing because of brain damage, we would expect the FA values to go down because there's more opportunity for water to diffuse in different directions. But it's not really diagnostic in the sense of what's actually causing those FA drops. So it is, in some sense, it can tell us something's changed about the white matter, but the danger is over-interpreting and making a strong conclusion that this is, white matter degeneration versus shearing versus some other change. And in principle, even, certain drugs or certain physiological conditions could cause greater permeability of myelin or some, other sorts of changes. So it's a measure. It tells you something about the white matter and how oriented the fibers are. It's just a little hand-wavy in terms of final interpretations. So there's other measures that we can get at this kind of voxel by voxel level. All of them could be argued to have some utility. I mean, they all have interpretability. So mean diffusivity is basically the amount of diffusion across all directions. So I'll get you in a sec, Leslie. So it's sort of summing the three principal eigenvalues or vectors. Relative anisotropy is the ratio between the dimensions, which is sort of like FA, so it's sort of how strongly oriented are things. And then the volume ratio is the product of those eigenvalues divided by the mean diffusivity. So you multiply them all together. So again, it's sort of a weird thing, but it's a ratio kind of measure. The criticism that I have is that sometimes I've read DTI papers where they report all this stuff, and it's kind of like, we got significant differences in RA in these areas, and we got significant differences in MD in some other areas, period. And you're kind of like, so what? And again, the problem is we don't really have strong interpretations at a physiological or a cellular level about what these things mean. So it's really nice that you can do the math and get numbers and apply stats to it. But it doesn't necessarily tell you anything super useful other than this population's brain is kind of different from some other population's brain. And hopefully you can do another study and replicate that someday. So we've talked about all these measures that are really voxel by voxel measures, right? So you can compute the FA or these other values at each individual voxel, and it tells you something about that voxel. Tractography is where we start to go sort of bigger and actually make inferences about where tracts are, so actual sort of pathways connecting different brain areas, which relies on that voxel by voxel data, but really requires you to go beyond and sort of connect the dots across voxels. And so the basic process is called streamlining, and it's pretty intuitive, right? So if you imagine this is a slice, so these are our individual voxels here, and the shape of the circle is showing its anisotropy. So the ones that are more oval are the ones that have more orientation to them. So with streamlining, you start at some point. So you define some brain area that you're interested in starting from and tracking where it projects to or from, because we can't tell the direction. But you take some point, it's an oriented voxel, and you figure out where is that one pointing to. So what's the next voxel adjacent to it that primary eigenvector is pointing most at, and draw a line into that next voxel, in that next one, you figure out where it's pointing to and just keep going like that. So you follow the orientation of the eigenvector in each individual voxel to connect the dots going through. You can see even from this simple example that even starting from two different voxels could actually take you in quite different directions. And so this is kind of a limitation of the technique, and often what's done is rather than starting with a single voxel, you might start from several voxels around there, like what they call a multi-start method, or do this multiple times in kind of a probabilistic way, because at some point, this makes it look fairly simple, but at some point, like even here, you can see that both of these lines are going through the same voxel, but they're going into different voxels after that. And so you don't necessarily have to rely on the direction of orientation of that single voxel, but sort of think about smoothing that line and think about where that was going before, where it's going next, and how much of a curve is going to be induced. So there are limitations because what you don't want to do is have your thing sort of going in circles or weird shapes. So often there's constraints that your line can only bend a certain amount per unit distance to constrain the tracts, which has both pluses and minuses because it can both help constrain things and make things not do weird things, but we also know that there are fibers in the brain that have fairly sharp bends. There are even fibers that are called U fibers because they're shaped like a U. And so you might have to tweak the parameters in the algorithm based on what you know about the tracts you're looking for to make them more or less sensitive to sharp returns. So you can do this in a couple of different ways. So you can start in the example before it started at one voxel or one area, and you can just sort of trace out from there where that goes. And that'll give you maps like that. And as I said, you would start from multiple voxels, see where they go, and usually a bunch within the same area will project in generally the same area. But a given area might actually project in different directions. The other thing you can do is point-to-point tracking. So that's rather than just defining a start point, you define a start point and an end point. So for example, we know that most areas of the brain, of the cortex, are connected to homologous areas in the other hemisphere through the corpus callosum. So you could say pick an area in the parietal lobe and the corresponding area in the left and the right parietal lobes, define those as your start and end points, and find all the fibers that track through both of those points. And so that would exclude all the ones that go in different directions. So if you have, and again, the connectome, there might be individual variability, but by and large, the connectome is genetically defined. And so we know where these white matter tracts are in general. And so we can have some pretty strong ideas that, okay, I want to look for this particular tract. I know that it passes through here, so we're going to help define it by defining both the start and the end points. So streamlining is that process, and then that gives us tractography, which is basically the tracts. This is an example, another way that you can do this rather than defining start and end points, because a start point sort of suggests that you're going in one direction. You can also define a midpoint and just find all the fibers that go through there. So corpus callosum is a good example of that, right? So you could define your seed in the corpus callosum and find all the fibers that pass through that, none really start or end there, right? So that'll go in sort of both directions. In this case, so this is an example study where they were looking at people with schizophrenia and characterizing differences in white matter connectivity in schizophrenia compared to healthy controls. And their regions of interest were based on areas that were already known to be affected by schizophrenia. In this case, this is actually a meta-analysis. Regardless, they looked at two areas, a left frontal region of interest and a left temporal region of interest, and defined all the tracts. And the different colors are showing you that given one particular region of interest, that area might actually be connected to several other areas. And so you've got tracts going in different directions, and they're color-coded based on sort of their pathways and where they're going. And so they were able to define which tracts might be affected in schizophrenia based on knowing that these two areas show differences, like even in fMRI kinds of studies. Then you can go and say, well, if they're showing different patterns of activation, do they also have different patterns of connectivity, or sort of less fibers going to particular brain areas or something like that. So that's kind of an example of how you'd use it. Now, some of the issues with tractography are that it really depends on having strong anisotropy at every voxel. So you start at one point, you're connecting your dots, but what happens if somewhere along the line you have a voxel that, for whatever reason, is highly isotropic instead of anisotropic? Your tracking is going to break down because you don't have a strong prediction about which direction to go. One way to deal with that is, again, you can sort of smooth out, so you can jump over one or two voxels and sort of guess based on where it was before, where it's going to go after. But one of the issues is that we know that, given that we're cutting the brain up into these voxels that might be 1 or 2 millimeters in size, there's going to be a lot of axons passing through a 1 millimeter cube voxel. And it's entirely possible, and in fact true, that fibers cross, right? So you might have different fibers going in different directions. They're going to cross through some of these voxels. And so you're not necessarily going to have, a strong anisotropic single direction of diffusion in these, because you actually have two different strong directions of diffusion. So that creates some uncertainty. And so you might end up with something like, this situation where you've got this is sort of a schematic one, because you're not likely going to find right angle bends, but it's just showing that you could have a voxel where you've got some fibers going in one direction and some turning in another direction, and your diffusion tensor is going to look something like that. So you've got one strong orientation of diffusion, but then a second direction that's relatively strongly oriented relative to being just sort of one strong sort of tube. And that gives you something like this. And so you can actually measure the uncertainty by looking again at the ratio of the eigenvalues in the first, second, and third. And that's what's being quantified by this representation, where we're taking a part of the brain that includes part of the corpus callosum, as it happens, zooming in on that. And what we see is where we've got this strong hourglass shape, that's indicating a higher level of uncertainty about the true direction of diffusion. Whereas where you've got the more sort of narrow hourglass shape, that's giving you a sense that there's very strong direction of diffusion. So in order to sort of address this issue of crossing fibers or noise or uncertainty, the first thing is coming up with a way of quantifying the uncertainty, which is looking at the ratio of eigenvectors. This is just an example showing you how different kinds of patterns of crossing fibers could affect your diffusion tensor. So the first couple are just, you've got a voxel where all the fibers are going in one direction. So that's pretty easy. You've got a strong V1 and a kind of weak V2. That's easy. Here you've got crossing fibers where there's equal numbers going in both directions. And that actually gives you basically a circle, right? So now you've got equal diffusivity in two directions. So in principle, it would be hard to distinguish that from like CSF or something where the diffusion is truly isotropic, even though it's actually got a very different interpretation. In E, you've got a more complicated or nuanced situation, because you've got crossing fibers, but you've got more fibers going in one direction than the second direction. So you've got a bit of an oval shape, but not as strongly ovaloid as here. So you might think, one way of interpreting that simply would be that there's just sort of less fibers or less strongly oriented fibers there, even though there's actually two populations crossing through there. These examples with, perpendicular fibers are pretty easy, but what if you have actually sort of more angled kind of crossing, or even multiple kinds of crossing here? The real point is that just looking at the V1, V2 ratio and trying to define a shape based on that isn't really sufficient to distinguish between these different cases. And so people have spent a considerable amount of energy on developing things like NOTI, that Leslie mentioned, and cue ball imaging, and these different approaches that try and start by quantifying the uncertainty, but then going further and using greater information, including this sort of larger scale rather than voxel by voxel information, to identify voxels where there might be crossing fibers and distinguish them from ones where there's no fibers or relatively low density of white matter fibers or something like that. But that's an important and inherent limitation of the technique, among others. This I already talked about, but it's just an example. Sorry, I think the slides got slightly jiggled out of order there. So this is point-to-point tracking, and I like this study because I do language stuff. But basically what they were looking at is Broca's area and Wernicke's area, which are in the left hemisphere are the two sort of core language areas, classical language areas. And it's known that there's a primary fiber tract that connects the two called the arcuate fasciculus, and this goes back to like Broca and Wernicke defining these things. And so, and this is, if you look at the date, 2005, so kind of early days in doing diffusion imaging. And they just wanted to demonstrate or sort of validate the DTI technique and use it to quantify the sort of size of the arcuate fasciculus. So here, what you're seeing, the top is just Broca's area defined, so top left is Broca's area, and I forget whether this is left or left is right, so neurological or radiological, but you see it, Broca's area, so the anterior frontal area in one hemisphere, then in the other hemisphere, and then on the right, Wernicke's area in the temporal-parietal junction in both. So those were defined as the seed regions, and they were doing point-to-point tracking, so which fibers pass through both of these areas. Gives you a map like this. And so what you're seeing here is this is a group level study. I haven't really, so this is the first time we're sort of going from individual to at the group level. So you would do this point-to-point tracking for an entire group of subjects, one by one, spatially normalize their brains, overlay them, and then the color map here is basically telling you how much overlap there is. So how many subjects showed fibers passing between these two regions at a particular voxel in the brain. And so the yellow is sort of the core areas of the arcuate fasciculus that you can see here. And you can see that, they define it pretty well. Interestingly, they also showed that, so the arcuate fasciculus kind of goes from Broca's area up above the temporal lobe through the parietal and then down, so kind of makes a bend into Wernicke's area. But they also showed that there were some fibers that went past that. And then there's a secondary pathway called the uncinate fasciculus that didn't come out as strongly, but they were showing that there were actually fiber tracts that were sort of going in this underhanded path as well. The uncinate fasciculus was already known, but this study actually helped highlight the fact that it was a secondary but important fiber tract connecting the two areas, and it wasn't just the arcuate fasciculus. So even though it started out as sort of a validation measure, and another thing I'm not showing is that they showed that there was much more yellow, so much more sort of overlap and more voxels that contain fibers connecting these two brain areas in the left hemisphere than in the right hemisphere, which is consistent with left hemisphere dominance for language. So it sort of validated the technique in showing things that were expected, but also provided some additional information as well that wasn't well studied. And in part, you can imagine that's because if you're doing this post-mortem in humans, doing dissections takes a lot of time. So people might focus on the primary tract that was already understood rather than sort of taking the time to find stuff or look for stuff that might not even be there. So that was kind of cool. An example of using single point tracking is another cool study from kind of the same era where they looked at projections from the thalamus to the cortex. So the thalamus is, we always talk about it as kind of the relay point between different sensory and subcortical inputs into the cortex. And we know that the thalamus actually has different nodes in it that project to different parts of the cortex. So you've got a chunk in the thalamus, and all of the cells from there would project to one part of the cerebral cortex. And so what they did was they took each voxel within the thalamus as a seed point and did the tractography to figure out where it was going. And color-coded back, so they figured out where it went, and if it went to the frontal lobes, they'd color-code that as blue, temporal lobes purple, occipital red, etc., etc. And so within the thalamus, you can see that they found that it wasn't sort of random, but in fact, all the areas that project to the frontal lobe sort of clustered together like this. All the areas that projected to the occipital lobe were back here. That's the lateral geniculate nucleus. And so again, it was a nice validation technique because we already knew within the human thalamus where these different geniculae were and where they projected to. So it was validating for this technique to show that in fact, these things lined up spatially with where we expected they were and that they clustered together so it wasn't totally random. And this is really important because some of the biggest limitations of DTI, as I've already sort of highlighted, are that it's very inferential, right? It's an indirect measure. We're looking at water diffusion, and there are principled reasons why water diffusion should respect white matter tracts, but it is secondary, and we do want to get validation of this technique rather than just saying, we think this is true, so let's do it and assume it's true and never actually check our assumptions. So studies like that, where you're validating and showing things that have been shown through other techniques, like dissections and animal studies, are really valuable in proving it. But at the same time, it's a structural technique, and it is inferential. And as I've already said, things like interpreting FA changes as changes in white matter integrity are inferential, and more work needs to be done to relate these things to one another. Largely, that's been done in animals, primates and other animals, where you can do diffusion imaging on them and then also do sort of gold standard injection tracer methods, sacrifice the animal, dissect it, and see how well those tracer methods respect or line up with the DTI measures that you get. But that's slow and painstaking work to do that, because typically studies would you don't want to inject lots of different brain areas unless you're using different tracers, because you need to be able to know we injected this kind of tracer in this brain area and it went to these 3 other areas or whatever. So it's very expensive, slow, intensive kind of work. Another thing you can do with this is integrate it with fMRI. So if you think back to when I was talking about functional connectivity and effective connectivity, a limitation of functional connectivity is that we don't know the direction that things are going in, and we don't even know if 2 areas that show functional connectivity, i.e. correlations in fMRI activity, actually are physically connected to each other, or if they're connected through some third area. So there's information going, we see functional connectivity between A and B. We don't know if that's a direct white matter connection between A and B, or it's A through area C to B, or if it's the thalamus is projecting to A and B sort of simultaneously. So if we include tractography and fMRI together, you can do things like take your fMRI hotspots and use those as seeds for your white matter tractography and figure out where those areas go to. And you can sort of map out things like that. One of the problems that we have with that, though, is that we rely on the anisotropy of the white matter to do the tractography. And so as soon as the white matter becomes gray matter, even sort of at the white matter gray matter border, our tractography breaks down because those voxels become highly isotropic instead of anisotropic. And that creates a degree of uncertainty because we don't really know where the fiber tracts end. They just sort of die off when we get to the cortex. And so we can sort of make assumptions that they just keep going straight into the cortex there, and that's not an unreasonable assumption. But it's also entirely possible that you have fibers that are going past an area of the cortex and just keep going. And it's a little hard to tell if something sort of goes alongside an area of cortex, if it's got branches that go into the gray matter there, or if it just bypasses them. So there's some uncertainty there, but it does help constrain things. And again, with the effect of connectivity, where you're looking at sort of the directionality of influence. Now, again, tractography can't tell you about directionality, but at least it can tell you if these areas are physically connected to each other or not. And that can help because, remember, effective connectivity and things like dynamic causal modeling are model-based techniques. So you come up with some model of how these different areas might be connected to each other and then test that. And so using DTI, you can actually constrain the types of models you're testing based on this anatomical information rather than just total guesswork. So there's studies that have done this. This is one that was looking at resting state functional connectivity. between different areas, and essentially showing that areas that showed functional connectivity, and that's sort of shown schematically there, also demonstrated structural connectivity. Largely, but not entirely. And so in particular, this medial prefrontal cortex to medial temporal lobe connection, which showed functional connectivity. So these two areas show correlated resting state activation. They don't actually have a physical connection between them, but they both connect to the posterior cingulate cortex and have functional connections. So it's suggesting that when we see that correlation between those two areas, it's not actually representing them being directly connected, but it's mediated by the posterior cingulate. So that's just a nice example of where the structural stuff can actually help inform our interpretations of the functional stuff. There's big work. The Human Connectome project, which I've mentioned before, that was the one that also generated the that parcellation of the brain based on all these different MRI measures. It's called the Human Connectome project because they're really trying to map out the human connectome using DTI, using large numbers of subjects, high resolution 7 Tesla imaging, and they have a website and you can actually go and access, because it's all open data, you can access their data, visualize it and stuff on there. So that's a pretty cool resource. And people are starting to push the limits again using high field imaging, and especially this has been done in either postmortem specimens or animal models rather than human models or postmortem animal models, because the issue again is to get higher resolution, high field scanners help, but you also longer scan times help. And so in this case, did I make a note here? Yeah, so these are from cadaver specimens that were scanned at 11 Tesla. So there aren't human 11 Tesla scanners, as far as I know, but there's very small bore ones that you can put mice in. And this was a 28-hour scan. So it helped that the animal was no longer alive because it's not moving. The cool thing is you get 92 micron resolution, so that's super high resolution. And so what you're seeing here is, rather than this sort of tractography where we're looking at white matter tracks between different brain areas, now they're looking at myeloarchitecture, which remember is like the fine-grained within looking across the layers of the cortex, horizontal fibers, vertical fibers, how are those connections going? And so you can see here, that's the B0 image is just sort of the structure showing you the cortex, the way it curves, you can see layers there. This highlights quite nicely. The red is the cortical layers, and then other layers underneath it. And the colors are coding for the directionality of the fibers. So you can see that in a very small area of space, you've got a lot of different fibers going in different directions. I'm not going to sort of go through all of this stuff, but then the other advantage of doing this in cadaver animals is you can also, once you've done the scan on the intact brain, you can slice the brain up, do the staining, and reconstruct that after you've done electron microscopy. You can reconstruct that and map it back onto the MR images. And so they're able to validate that, when they're seeing these orientations, it lines up with what you see in sort of standard myeloarchitectonic staining kinds of methods. And get, just like I talked about with the human, the Ulick brain project, where they're looking at post-mortem probabilistic maps of cytoarchitecture. You can do the same sort of thing here and get profiles across the cortex of the myeloarchitecture and start defining that's a hard word to say, myeloarchitectonically defined areas based on these gray matter, or the FA profiles, and how they change along the cortex like that. So that's relatively recent. That's a 2015 paper, but that's sort of the extreme end of pushing the resolution and capabilities of this technique. Not practical in humans, but still very cool and very informative. So I guess I'm like, where'd that slide come from? The point is just that they're doing this in the cortex. And so what I said before, like DTI breaks down when you get to the cortex. With high enough resolution imaging methods, that actually isn't necessarily true. It's just that our sensitivity is relatively low in the cortex, and so you need higher field scanners, different processing techniques, longer scan times, and so on in order to get that. So it's not an inherent limitation of the gray matter, it's just the technique and how far you can push it. All right, so that's the end. Thanks very much. We're actually caught up. It's amazing. Next lecture I can start as that lecture. All right, so yeah, if you're able to make it tomorrow, Friday, on Friday I'll see you in the computer lab. If not, hopefully your group can help you out, and Liam and I are available to meet with you if you get stuck and stuff like that. So I'll see you then.
So I'm going to try to give work away.
Good. I mean, as far as surgery goes, kind of fun.
Yeah.